Hello, Isa Ssekandi is my name. Today we are looking at a group of organisms called the bacteria. Bacteria fall under a group of organisms called prokaryotes. This comes from two Greek words, pro meaning before, and here we have karyota meaning nucleus. In simple terms, these organisms have a primitive or less developed or underdeveloped nucleus. Their nucleus doesn't have a nuclear envelope, and several organelles in that in these organisms don't have membrane bound. They are not membrane bound. So we proceed by looking at the origin of these bacteria. Several scientists work tirelessly to look at their origin and to give us an insight into what these organisms were when they were first discovered. So the very first scientist was Antoine van Leeuwenhoek. Antoine van Leeuwenhoek. So this is a Dutch scientist who came to realize that these bacteria looked like small single-celled organisms. At that level, they were within the rainwater that he observed using his primitive microscope, wooden microscope, which was single-lensed. So he observed them as small molecules, and some of them could move within the water, and he named them animalcules. That was in 1667. A few years later, that was in 1837, a German biologist, that is Gottfried, came in and coined the term bacteria. So he gave them the name bacteria. Actually, Gottfried was able to realize that these bacteria were like small sticks. They were like small sticks within the microscope when he analyzed them. So they looked like small sticks, and in German the word bacteria means little sticks. Little sticks means little sticks. The other scientist was Louis Pasteur. This was a brilliant French scientist who used broth. Broth is like a culture mixture, growing medium of meat, some cereals, and other material that can support microbial growth. So he used boiled broth. He left in two flasks. One was covered and one was not covered. He realized that the uncovered flask, some organisms were able to grow, microbes were able to grow in that flask. At that level, he didn't know that they are bacteria, and he realized that they are within the air, and that's why they were able to enter into the flasks. And the year was 1867, a few years after Gottfried's research. So at this level, research was showing that in the air and in very many places around, there are small, tiny unicellular organisms that are known as bacteria, and they are within the air, they are within water, they are within very many materials. So little did they know about their structure. So later on, research showed us that the structure of bacteria, a typical bacterium or a typical bacterial cell takes up this structure. Some of them have flagella, which is small, have flagellum. Some have and some don't have. So free-living bacteria that are found within water, within ponds, within in free surfaces, are the ones that like to have flagella. Some bacteria are multiflagellated. They are multiflagellated. We have 1, 2, 3, 4, several flagella, Others are uniflagellated, others are diflagellated, so the forms are so many. However, we need to know that bacteria that cause disease that are found as parasites within different organisms may not have the flagella. Then some bacteria are covered by a tough capsule outside, which is a slime layer, sometimes called a slime layer. So that slime layer enables bacteria to survive harsh conditions and also stick together within colonies. 2, we do have what we call pili. Pili is shorter than flagella, they are shorter. Another name for pili, pilus. Another name is fimbula. Fimbuli, when they are many. When they are many, we call them fimbuli. We do have a cell membrane. They do have a cell membrane inside, and then a cell wall. So there's a cell membrane and a cell wall. A cell wall. The cell membrane has inifoldings. This is the photosynthetic lamella. This one is found associated with bacterial chlorophyll and other photosynthetic pigments. Another inifolding is the mesosome, concerned with synthesis of energy, that is ATP. Here is the cytoplasm of the bacterial cell. Here is the DNA, which is found in the nuclear region. So what you can see here is the nucleoid region carrying DNA of the bacteria. These are the tiny spherical structures that are concerned with protein synthesis. We call 70S ribosomes. 70S in the sense that they are smaller than the 80S of eukaryotic cells. This is a food vacuole for this bacterial cell, the food vacuole. And then we do have plasmid DNA. Plasmid. Another name for plasmid DNA is extrachromosomal DNA, meaning you can call the nucleoid DNA as the chromosomal DNA. So the chromosomal DNA is smaller. The DNA of this bacteria is smaller than the DNA of the eukaryotic cells. Actually, this DNA carries close to 4,000 genes compared to DNA of the eukaryotic cells that carries close to 10,000 genes. So extrachromosomal DNA, the plasmid DNA, is concerned with carrying genes that are important in antibiotic resistance, in antibiotic resistance, resistance to drugs, and also resistance to harsh weather conditions, and also genes that are important in conjugation and reproduction. So that's the structure of the bacteria. We need to note that in bacteria that are found in harsh weather conditions, for example, bacteria that inhabit hot springs, water whose temperatures usually go beyond 70, or sometimes beyond even 100. We all know that hot springs temperatures can go as high as 150 degrees Celsius. So bacteria that inhabit such places usually have a capsule to protect themselves from such harsh conditions. Thank you so much, and that's the structure of the DNA. Stay tuned in for the next episode concerning the next portion about bacteria.